I'll give everybody a chance to make sure their connections are moving through here.
You can see me now. Sorry, I wasn't expecting to be on video. Here we are.
Yep, I can see you now.
Great. Hi, everybody.
Hi. So on behalf of the 9P minus research team at Washington University in St. Louis, I'd like to welcome all members of the Chromosome 9 community to our webinar. Tonight we'll have a chance to review the research project which we here at Washington University in St. Louis are working on, specifically on 9P individuals. We'll be able to hear a little bit how far we've gotten so far and how well we're doing. And we're anxious to have questions and ideas and partnerships with all members of the Chromosome 9 community, especially those 9P minus members. We have a great group of members of the research team here with us this evening who will review different parts of our research project. And we welcome any questions about the project, about strategies to get involved in the project, about ways you can help with the project, because we really need the partnership of all members of the Chromosome 9P community to move this project forward. I received today an email from a mom in Spain who has a 9P minus son who's anxious to participate. And she reminded me that we have 9P community members all around the world. So let me start by asking members of our research team to introduce themselves and say a bit about what they're doing, and then we'll move on to talk a little bit more about the specifics of the project. So Dr. Turner, why don't you start?
Thank you. Yes, I'm Tishelle Turner, and I'm an assistant professor here at Washington University, and I am the genomics lead on the 9P minus project, and I'll be giving you updates about that tonight.
Great. Thank you very much. Dr. Mitra.
Hi, I'm Rob Mitra. I'm a professor in the genetics department at Washington University. And I'm working to set up pipelines so that we can understand once we have a list of genes from the breakpoint analysis that Tishelle is doing. I'm working to set up methods so that we can understand exactly what these genes do and how they cause their phenotypes.
Great. And Ms. Tate.
Hi, my name is Tori. I'm the clinical research coordinator for the group. I talk to everyone in our group. I facilitate the research with everyone, and I'm the point of contact if you're interested in joining. And really, I'm with you every step of the way, from the time of the sample collection, virtually, of course, to the time that I collect the questionnaire and medical release form.
Great. Yes, Tori is your point person. I'm also happy to serve as a point person as well. But if you really want to get something done, I would email Tori. She's like the brains of the operation here. We're also joined tonight on the panel by Ms. Sierra Dickson and Mr. Leon Eidelman, both members, parents of individuals with chromosome 9P issues, and they have been unbelievably supportive and helpful in terms of both putting together our research project and also in helping to make the research project go, and hopefully to engage as many of the 9P community members as we can possibly engage. We're also going to hear tonight, I hope, from Dr. Patricia Dickson, who is the chief of the Division of Genetics and Genomics here at Washington University in St. Louis. I know she was going to join us, and I'm optimistic that she'll be here in the not too distant future. So without further ado, why don't we start, Tori? Do you mind just giving them the numbers a little bit about sort of where we are with recruitment, and then we'll go through what the study is about and entails.
Yeah, of course, I'd love to. So the study did start in 2017, and since then to date, we have 117 individuals. That does include individuals who have given saliva, as well as our current protocol with the blood draw. But really just focusing on our newer protocol since last year, we have enrolled 42 individuals. And with those individuals, we have received 32 of our kits for the blood collection samples. And we're kind of across the board for collecting information, the phenotypic information for the clinical findings, but we have really tried to make that as easy as possible for our participants by creating a link to a secure website that we use at Washington University that's approved for it, for that questionnaire to be filled out at your leisure. So it's a little bit easier than the paper form, and it's become more comprehensive with time as well. And I work every day to reach out to the medical groups. They've been really supportive in sending me as much information, but that's an ever working process.
Great. That sounds really great. And we'll hopefully talk in a minute a little bit more about the process of actually enrolling in the study. Let me say that for all of the Chromosome 9 community, please put any questions that you have or comments that you have into the chat or the Q&A, and we'll get to them by the time we, as we go along during the webinar. We're also very pleased to be joined by Dr. Patricia Dickson, the Chief of Genetics and Genomics at Washington University in St. Louis. And Dr. Dickson's going to introduce herself briefly and tell a little bit about what she does on the research team.
Hi, thank you so much, Dr. Cole. So yeah, I'm Patty Dickson. I'm a professor of pediatrics and chief of the Division of Genetics and Genomic Medicine. So I'm a medical geneticist, and I lead the phenotyping portion of the research team. So we are geared toward collecting medical records and phenotype information on our participants, as well as working out logistics of specimen collection, enrollment, and any other interactions and communications.
Great. And I think, as I think everybody understands on the call, our research project is really aimed at using state-of-the-art strategies to identify exactly where the 9p breakage points are, and down to much greater resolution than has been previously available. And Dr. Turner will talk about that in a minute. And then try to understand how the characteristics of each individual with 9p are associated with the specific areas of the 9p chromosome that are either deleted or changed or duplicated. There's a question in the chat about what made you choose chromosome 9p? So that's an interesting question. I would say that I can tell you my personal journey, and maybe others can sort of chime in here. My personal journey in terms of choosing chromosome 9p is because I have been involved in the care of a patient for the last 19 years with 9p minus. And she and her family are terrific. And I became increasingly aware of the lack of research focused on 9p minus. You'll hear from Dr. Turner about why research is so valuable and potentially discovery oriented when she talks. But in partnership with with members of the chromosome 9p minus community, we started several years ago to try to begin to develop some kind of a research approach or project for 9p minus individuals, and we have made a tremendous amount of progress, I think, as you'll hear about this evening. So thank you very much for that question. Dr. Dickson, do you want to say just a word more about the phenotyping and the possibility of using telehealth for phenotyping?
Sure. We are collecting, I mean, as medical geneticists, we collect phenotype information as a matter of course. And so we are doing this by standard ways of looking through the medical record, having patient questionnaire. We are able to perform some telehealth assessments, and we're looking into ways to do some behavioral assessments. Not sure if Dr. Cole, if this was what you were thinking
Yes.
of talking about. Okay, working with the Intellectual and Developmental Disabilities Research Center to try to offer some telehealth assessments that of behavior through for individuals in the study. So this is something we're looking to potentially do in the future. But generally we are cataloging phenotype as is pretty standard for most of these studies. The unique part of this particular study is really the genomic sequencing technologies and some of the functional studies that are done downstream of this.
Right. So I think it is important to understand that we are trying to collect information about each individual who participates in the study that's standardized and specific. So as members of the 9P community might understand, there are characteristics of 9P individuals that seem to be relatively common, and then there are other individuals seem to have different characteristics, and we're wondering whether we can learn what genes are associated with which particular characteristics by collecting the information that Dr. Dixon has just outlined. And I know it's a pain in the neck because chromosome 9P individuals have a lot of information in their medical records. But it's very important that we try to collect that information in a standardized format so that we can make the kinds of comparisons and associations that are vital for our being able to try to begin to identify specific genes associated with specific 9P characteristics. There's a question in the chat about, are you focused on pediatrics only or adults included? Adults are definitely included. We want to have anybody in the 9P community. Please don't, Dr. Dixon and I are both sort of oriented towards children in our clinical practices, but we also see adults, and we would be pleased and honored to have members of the 9P adult community also join our study. So there's a question, Dr. Dixon, about from the Q&A, how rare is 9P minus? Do you want to take a stab at that?
I think it's not incredibly rare. I think it's estimated somewhere around 1 in 50,000 individuals. So it's not for a medical geneticist, it's not exceedingly rare. It's probably something, in all of the what we call chromosomal microdeletions and microduplications are much more common than we anticipated. And as technology improves and is more widely deployed, will probably be even more common than we previously thought.
Great. So what it means to be enrolled and participate in this research study is that you contact Tori, and she goes over with you the informed consent form. And what the consent form does is it allows us to get from you, first of all, medical records and medical information about the 9P individual. And then our study also requires that the 9P person who's being studied, who's participating, has a sample of blood drawn in special tubes and sent to us. And we will send you or the provider or physician who is going to help us with the blood draw. We'll send you the tubes, the instructions, a return, paid FedEx envelope, the whole 9 yards. But you can't just go to your local hospital and say, draw some blood from me, please. You have to receive a kit from us after you have given consent to have blood drawn and given consent to send us your medical information. So once we receive the blood sample, then Dr. Turner goes to town and Dr. Mitra goes to town on beginning to analyze that blood sample. And Dr. Turner is going to tell us a little bit about her progress to date on some of the samples that she's analyzed.
Okay, shall I go ahead now?
Yes, that'd be great.
All right, I just have some slides here tonight. Okay, so thanks everybody for coming, and it's nice to virtually be here. And as I mentioned earlier, I'm the genomics lead for the 9p- project. And really my goal is, since I got into it, my goal really is to get to precision genomics for precision medicine in 9p- syndrome. And, you know, working with this incredible team here at WashU, we're covering many aspects of that. But tonight I'm going to focus on the genomics. And I thought I would give you a quick background on what this really means, because I've been reflecting on it for a while now. And precision medicine itself is a concept that's been around for a few years. And the idea is that we can ultimately tailor treatment, therapeutics, and healthcare to each individual. For me as a human geneticist, it's really quite an exciting area, because a key feature is that we really want to understand the individual's genome, because the variation in the genome ultimately has an impact on phenotypic features, predisposition for disease, and response to treatment. And so ultimately, I think that we need precision genomics if we're going to do precision medicine. And really, until about maybe 2 to 3 years ago, we couldn't do precision genomics, but now we can. And what does that actually mean? To me, precision genomics in 9p- means that we understand exactly where the breakpoints are of the deletion and other often complex structural variants, that we can look at variation on the chromosome 9 that does not have a deletion, and that we can also look at variation around the rest of the genome, because this could be very important for the individual. So we actually, as a team, have been working quite hard to understand all that we can about 9p-, and we've recently put together a paper where we've gone back and looked at all of the phenotypes seen in the literature. And this table, I know it's quite small, but I just wanted to give you an idea that when we look at the percent of individuals with a given phenotype, it's quite variable in 9p-, but yet there is a shared phenotype of developmental delay. So there is a path forward that we could, you know, understand some of this kind of variation and developmental delay in particular. And so, as you are probably well aware, 9p- is typified by having a deletion somewhere on chromosome 9. So if we look at chromosome 9 here, we have 2 copies, right? And with a deletion, a small portion of that chromosome is missing on the P arm. Now, traditionally, this has been diagnosed by 2 different technologies. One called the karyotype, where we can actually see the chromosomes as shown here. So here's a 9 without deletion, and here's a 9 that has a deletion. Or by array, and that's intensity based. So you can see that these values by and large are lower than these. And so that's a deletion region. Now, these technologies are pretty cool, and they have revolutionized the field, but they are imprecise, meaning we can't get to the exact breakpoint location like we can do with newer technologies. Okay, so we actually have aggregated from the literature what is seen in individuals with 9p- based on karyotypes and arrays. And you can see actually there are many different deletion sizes shown in red. So this is just the P arm of chromosome 9, and each of these bars is a different person. And as I mentioned, red is deletion. What we also notice beyond the variability in size and location is that some individuals also carry a duplication, which is shown in blue here. Okay, so this is great that we know this, but we actually want to go to the next level and understand what the precise variation looks like, and couple that with phenotype information. So as mentioned just a few minutes ago, we can really go in and understand 9p minus from a holistic perspective. Like we can understand the whole of it because we can pair the phenotype and the genotype. Now, there's one other consideration we can make with 9p minus, that it's been documented that about half of the time, individuals with 9p minus also have a translocation. So they will have a deletion, as shown on this chromosome, but then they have another piece of another chromosome that attaches to that one. And this has actually been somewhat difficult for genomic technologies to detect. And so we can actually do this now with sequencing and resolve the full variation. So in June of last year, we published our very first paper on 9p minus, where we actually did do just that. We understood exactly where the variation was, the deletion here. So this is just a schematic of chromosome 9. And this little red piece is where the deletion was. And then this purple piece is another chromosome, in this case, a piece of chromosome 13. And then the rest of the chromosome is at a typical copy number of 2. And we could actually find out exactly where the breakpoint occurred for the translocation, which I'm showing here. And importantly, on the chromosome 9 side, which is down on the bottom, you can see that this breakpoint doesn't break any genes. And if we had used an array, perhaps we would have not quite got the breakpoint right, and we might have inadvertently found that this gene was disrupted or this one. And that can actually have a big impact on the statistics that we do downstream to understand phenotype, genotype correlation. So knowing these precise breakpoints is important. And we also did further characterization of the deletion. So as I mentioned, we want to get to this precision genomics for precision medicine in 9p, which I would call PGPM. And this is really, I think, pretty cool that tonight I can show you the genomic results for the first 20 individuals who have enrolled in our study. And so maybe some of these individuals are on the call tonight. And so we are working very hard at getting the samples and processing them through our genomic workflows. And you can again see that in red, there are different sizes of deletion. And in fact, 2 of the individuals also contained a duplication. So individual 1 had a deletion and a duplication. Individual 13 also had a deletion and a duplication. So we're just really beginning to understand this variation. And we also, along the way, have been able to resolve individuals who have translocations in addition to the deletion. So in this particular case at the top, the individual had an entire chromosome 14 attached to a chromosome 9, which had a small deletion. And then it had a small piece that actually had moved to another chromosome, and then another deletion, and then a mosaic duplication. And the remainder of the chromosome was at copy number of 2. Without precision genomics, we would not, and this is in the newer sequencing technologies, we would not have been able to resolve this. We tried using the older technologies, and it wasn't until the newest technologies available that we could really understand this. That's why we think it's important that at this time point in history, this is the time that we can really understand 9p minus from the genomic perspective. So there's 2 other individuals I wanted to highlight. So one that has a translocation of 13, and then another one who has a translocation of a piece of chromosome 2, and then their 9p deletion. So this is where we're at right now. I mean, it's still early days, right? So our ultimate goal here is to finalize the precision genomic workups for these 20 individuals. So we're beginning to go back and look at the remainder of the genome in those individuals, as well as the other chromosome 9. We ultimately want to get to 100 individuals with this technology, because at that point, we think we have the statistical power to begin to really look at the phenotype genotype correlations. And of course, we want to integrate this with the larger working group. And I want to point out that although I'm speaking today, I'm speaking on behalf of a large network of individuals here at WashU, both from my lab, the Genome Institute, and WashU more broadly. So when I present this, it's not just my work, it's the work of our larger group. And I'm so delighted to be a part of this and to show you where we're at today. So I will stop there and thank you.
Great, Tishelle, that was terrific. I think it's for those 9P minus community members who are on the call, Dr. Turner's work is really like going from flying at 50,000 feet and trying to see whether there's like water or trees or desert down there, down to about 10 feet. And so you can really distinguish what the specific locations are of these differences in what parts of the 9P chromosome were deleted, duplicated, or what other chromosomes were translocated. Tori, we have to figure out, we've had a request to be sure that we get the slides that Dr. Turner showed and any that Dr. Mitra may show, and we need to put them someplace where people can get at them. We'll try to figure out how to either email them to the registrants or put them in a place where the registrants can access them if they want them.
Okay.
Okay. There are a couple of other comments in the chat before we get to Dr. Mitra. Let's see. One question is about our adults included. Yes, as I answered that one. Then a clinical outlook for your daughter's future. Does participation in the project allow for us to have access to findings? Yes, it allows you to have access to findings, but remember, these are research findings. So these are not findings that you can use today or tomorrow to make any definitive decisions, for example, about therapy for a person with 9P minus. But they do, we will be able to share the findings that Dr. Turner has described for individuals with the individual patient or family who is affected. Okay, we sent in our blood draw about 9 months ago, and we're working on the questionnaire. Are there any preliminary findings? Yes, there are. And as I said, I think you saw a little bit about the first 20 anyway from Dr. Turner. You're going to hear a bit from Dr. Mitra. Okay, let's see, two other questions here. Will we get a record of the results for our individual as well? We will describe the results, but as I indicated, it's important to understand that the results aren't going to be able to be used to say, okay, fine, if we start this medicine tomorrow, the characteristics of 9P minus that the individual has will go away. That's not going to be possible. The study is likely limited to those in the US since a blood sample needs to be mailed. Is this correct, or can individuals from Canada participate? Yes, we think individuals from Canada can participate. The major issue is trying to find a place where we can help you get your blood drawn. We are in the process. We've worked hard. And I'm not sure whether there's a family who will be able to sort of talk a little bit about that in a minute, but we've worked hard to try to find places that 9P minus individuals and families feel safe in going to have their blood drawn. And we know that this is a big speed bump to try to get over, but we want to try to help you with it. And remember, after you give consent to participate in the study, you need to receive a kit from us that will contain instructions and the blood tubes. Because if the blood arrives in the wrong tubes, then what Dr. Turner just described and what Dr. Mitra is about to describe is not going to be possible. Okay, Dr. Mitra, do you want to talk a little bit about what happens after Dr. Turner gets finished pumping genes through her pipeline there?
Yeah, sure. I would love to. Let's see here. Can you see my screen? Or is it blank? Uh-oh. That's interesting. Can you see my screen?
Yes.
Yes. Okay. I'm just going to go flip through the slides because when I try to do presenter view, it seems my computer seems to have a little bit of trouble. So right now, we, as Dr. Turner just explained, we're very focused on getting to 100 patients and mapping the breakpoints from these 100 patients so that we can correlate the different sets of genes that are in different patients with the phenotypes. Okay, and that's largely what we're focused on right now. And we're very fortunate to have a really superb clinical team, Dr. Cole, Ms. Tate, Dr. Dixon, and then we're just incredibly lucky to have Dr. Turner, who's a world-class computational biologist, and she's particularly focused on this problem. Her expertise is particularly on these types of complex rearrangements and collecting the genomic data. So that's really where the main focus is right now. And what we expect is that by looking at these different sets of genes and correlating them with phenotype, we'll find genes that are associated with different traits, okay? And that in itself, we expect will be very useful, will be very illuminating, okay? But then, you know, once we say, for example, find a gene that's correlated with a particular phenotype, what we'd really like to do next then is start to understand the mechanism by which this happens, the particular molecular pathways that are active to cause this phenotype, and even just what cell type is this gene acting through, because it could be acting through, there's a variety, you know, there's hundreds of cell types in the body, and it could be any particular cell type. So what we'd like to do is understand precisely in a molecular sense how these genes are working, okay? And so what I'd like to do now is just give you a flavor for how we're gonna try to attack this problem, which is really, again, going to get into full swing when we get the full 100 patients and then we map those genes, okay? And so how do we know what a gene does, or how do we know molecularly how a gene acts? And the way we do it, our toolkit for doing this is largely based on a very simple idea, which is we take a cell or we take an organism like a mouse and we break that gene in the cell or we break that gene in the mouse and we look for resulting phenotypes, okay? And we characterize it molecularly with a large number of molecular measurements. And that gives us clues as to what that gene is doing, okay? And so in a sense, we can break that gene in two ways. We can turn it off, we can knock it down or turn the levels of the amount of protein that's made from that gene down, or we can overexpress it so that large amounts of that protein are being made. And then we look and we see what happens, okay? And our tool of choice for doing this is a technology that's called Crispr. So when you knock it down, it's called CRISPR-I, that's here on the left. And when you turn it up, it's called CRISPR-A, okay? And you may have heard of CRISPR. It's called Cas9 or CRISPR, which are sort of synonyms for the same technology. And it's made a lot of buzz in the popular press. The way to think about CRISPR is that in some sense, it's like a molecular smart missile. Okay, we can program this protein to go to any place in the genome very simply and very easily. Okay, and then from there, by making a few modifications to this protein, we can get it to turn genes on or turn genes off. Okay, so this is really our workhorse. So once we have a list of genes that we want to turn on or turn off, we can then target them with these sort of Cas9 CRISPR technology, and we can turn them on and turn them off very relatively easily and at a high throughput scale. And then we can do a large number of molecular characterizations. So we can measure the global RNA levels throughout the whole cell to see which genes are upregulated and which genes are downregulated as a result of our initial perturbation, okay? And we can also look at cellular morphology and cellular phenotype. So that's our main philosophy of how we're going to try to understand how these genes actually act. Okay, but it's actually, you know, this sounds very easy, and actually this is really a miraculous technology, and it's greatly accelerated our efforts. But it's actually a little bit harder than you might think, because for example, we need to find the right model to work in. So we could knock a gene down, or we can turn a gene up, but what cell type should we use? Should we use neurons if we're looking at a delay phenotype, at a cognitive delay phenotype? Should we use oligodendrocytes? Should we use, so that's a very complicated question. We don't know when this particular gene is acting. It could be acting early in development, or it could be something that acts throughout the lifetime of a patient. And then furthermore, you don't necessarily know what to look for. So one way to kind of to sort of figure out how a particular gene works is actually to make it. This sounds counterintuitive, but rather than just look at a small number of genes, sometimes it's helpful to actually look at a large number of genes, okay, and then make a very large number of molecular measurements. And then what happens is that genes that tend to act in the same pathways tend to give particular molecular signatures, and then they can be grouped together by these signatures. And that allows you to take your one gene, which you didn't know very much about, and connect it to other genes where you maybe know a lot about, or they all make the same molecular signature, they all have the same molecular signature, and now you can study this whole set and make a lot more progress. Okay, is that clear? So that's, in some sense, that's really our strategy, we've set up this pipeline where we can take a large number of genes that are involved in, we're particularly interested in developmental delay and in developmental delay and cognitive pathologies. So we can take a large number of genes that are involved in this set of phenotypes. We can make perturbations in very high throughput in a large number of cells. So what we can do here is we can take fibroblasts from the skin and we can convert them to a large number of different cell types, so neurons or oligodendrocytes or glia. So that would be different cell types in the brain. But we can also make other cell types as well for other phenotypes of interest. And then what we can do is we can make these perturbations on a large number of genes, hundreds of genes at once. We can make measurements, so we can image the cells. We can image the cells. We can also make molecular measurements, so we can say which genes are upregulated and downregulated in response to the original perturbation. And we can also look at we can look at the structure of the genome, or chromatin structure, okay? And by looking at these molecular measurements, we can group these genes into pathways, and we can start to learn more about what these genes do. So this sounds very complicated, but let me just give you an example with a smaller number of genes from a pilot that we performed that actually gave some nice results, and it'll just give you a flavor for the types of results that we hope to achieve with the gene list that Tichelle comes up with from the 9P project. So we did a little pilot where we took a lot of genes that are involved in autism, that have been implicated in autism, but it's very unclear what these genes do. And what we did is we made perturbations to all of these genes, and we did it in a system where we took cells that were neural progenitor cells and they differentiated into neurons. Okay, and then what we did is we made a large number of molecular measurements on these cells. Okay. And what we could see by looking for these molecular signatures and grouping these genes that we perturbed together, they actually fell into two groups. One group of 4 genes that delayed differentiation, and another group that accelerated differentiation, okay? And in fact, so what that allowed us to do is allow us to, one, group these genes together, and 2, assign some sort of molecular function to these genes, okay? And so what was really interesting is that what we found is that, for example, the genes that delayed differentiation had a large number of different phenotypes. So they tended to proliferate slower, that they tended to divide not as quickly. They tended to have, the neurons did not send out processes as effectively, okay? And what was really interesting is that these molecular phenotypes were actually well correlated to patient phenotypes. So for example, the patients that had genes with mutations from this delayed differentiation module tended to have more microcephaly, which actually makes a lot of sense because they had a reduced amount of proliferation, okay? But in contrast with the genes that accelerated differentiation, they had an enhanced proliferation and they had a degree of macrocephaly. And what we could see is that there was a very good correlation between our molecular implications and the actual phenotypes that we observed in the patients, okay? So this is what we would hope to achieve very similarly with the 9p minus. We do very similar analyses, and then by layering in onto other sets of genes that we're analyzing, we can group them into modules just like we did here, and then hopefully correlate these molecular processes with actual patient phenotypes. Okay. So right now we're scaling up to more genes. Most of our work to date has been on knocking down genes. We're working to turn them up. And then as we begin to get a very high confidence gene list from the 9p, we're ready to go and plug those genes right into our libraries. All right, so I'll just end there. Thank you.
Great, that was terrific. So I think what you heard between Dr. Turner and Dr. Mitra is that Dr. Turner identifies what the genes are that are affected in a 9p individual, and Dr. Mitra tries to figure out what those genes do or don't do. And then once those two sort of discoveries are made, we hope that there will be a path to understanding how to address the gene problem in individuals. That's the sort of long-term goal. We're not there yet. We're not close to being there yet, but I think we certainly have made more progress in the last year or two than I have seen taking care of patients with 9p minus in the last 19 years. And I would defer to Dr. Dixon, who's probably taken care of a lot more of these patients than I have in terms of the kind of progress that's being made here.
Yeah, I mean, I would say in an answer to one of the questions in the Q&A too about, what does this mean for the future? And we have a good research plan to try to map and connect the genomic variation and the copy with the phenotype in individuals. We have a cellular model plan, as well as going back into the patient phenotypes and trying to relate those with the cellular models. We don't know. We're going to make discoveries. And I would say the history of medical discoveries, these types of experiments where you better define and map regions definitely do help the field and science move forward. So we definitely hope that we can make some progress here. And I think that we have a very good plan. We've made a lot of progress in collecting the information and samples and doing some of the sequencing so far. So as we get our data set moving forward, I'd say that we have a very well-organized team and are making very good progress on this project.
So one of the in chat asked, Patty, as a layperson, is there glue that has joined the area on chromosome 9p that is deleted? How does it communicate with the rest of the chromosome? So basically explain how a deletion works.
Yeah, I mean, DNA has sticky ends, right? So they join back together. And this is one of the features of DNA that allows our genes to recombine and us to be different from each other. But it's also, can unfortunately cause copy number variations that and other mistakes that can happen as the or just variation that can happen as things recombine. And so, yeah, it's a feature of DNA that it has these sticky ends and polymerases and other enzymes that heal the nicks that are made and the cuts that are made in DNA so that it's a continuous strand.
Great. I'm just replying to a participant from Merida, Venezuela in Latin America. And I've suggested that she reach out to you, Tori, to get a little bit more information about participation in the study. Okay. Okay. I don't know if Miss Darlene Barents is in the among the participants. If she is, Mr. Bauer, could you see if she would be willing to talk very briefly about her experience in participating in the study? Can you,
I'm not seeing, what's her name?
Barents? She's the mother of Sandra Sissons. If not, we can sort of move forward here.
Darlene?
Yes, Darlene Barents. Oh, Ms. Barents, there you are. Good evening.
She's muted.
Can you unmute yourself?
I've asked her to unmute.
Tori, can you put your email in the chat and the Q&A? Yes, Ms. Barents, thank you very much for joining us this evening and for being willing to share your experiences. You want to say a word about where you are? What part of the country?
Oh, me?
Yes.
Sure. Hi, I'm Darlene Barents. I'm the mother of Sandra Sisson, who is 9P minus. She's 41 years old. I'm in Southern California, Highland, California. And Tori asked me if I would just tell briefly about our experience with the study and so forth and the procedures. And I just want to tell all the 9P minus families that this is so important. And it's really just 3 steps, at least so far. We received the, it's called the informed consent document, which Sandra signed. And then I, as her conservator, also signed and sent back. And then we received a questionnaire that was it had some difficult parts for me to answer. Sandra's 41. Those of you with younger children probably remember. It was a lot for me to remember, 41 years. But I went back into her medical records and so forth and was able to fill it out to the best of my ability. And there were some words in there, medical terminology, that was pretty difficult for me to understand. So I Googled some of the terminology and even asked Alexa. She didn't know a whole lot, but Tori helped me with that. Anyone that's working on this, Tori has been such a wonderful help. And she's always so upbeat and happy to hear from you and helpful. Don't be embarrassed to call her and say, I don't understand what this is, because she won't make you feel dumb or anything. Anyway, the questionnaire was quite lengthy, but I think it's, definitely necessary for the study. So we got through that with the help of Tori. And when you are working on that, you probably will have to refer to some of the medical records in the past and your memory. So I happened to send Tori more than she even asked for. The 9P minus families, since before Sandra was even involved, have sent stories each year of their child and what their just what they've done for the year, their milestones and so forth. And I sent Tori Sandra's whole packet of her whole life, just in case I missed something in the questionnaire. But we got through the questionnaire. They sent the blood kit, and it was just so convenient because the company that was doing the blood draw came out to our house. So we didn't even have to go to a hospital or a clinic or anything. It was great. And they're the ones that sent the blood work back in. We didn't have to do anything except Sandra sit there and get her blood drawn. So it was convenient and it was great. And I just want to tell the families how important this is. I'm sure you do know it, but having a child that old, well, an adult, when Sandra was diagnosed, I was told to institutionalize her, that she would be a vegetable. She'd never walk or talk, and that, you know, I wouldn't be able to deal with it. Well, I didn't do that, of course. But Sandra, anyway, I just wanted to say that wouldn't it be wonderful when a child is diagnosed and the doctor can tell the parents something? I mean, you know, we were told nothing other than really negative things. But with this study, with what all of the doctors and researchers are working on, you know, in the future, someone born with 9p minus or diagnosed with it, you know, the parents may be able to be told, given some hope and actually some information on what the child might really develop, you know, which parts, whatever. Anyway, I think this research is just absolutely fantastic. I am so impressed with the work that you guys have done. And I just want everybody to know it's really easy to enroll and be a part of it. Thank you.
Great, Ms. Behrens, we really, really appreciate your perspective. Let me just add that for 9P individuals who are participating in the study and who are less than about, I guess, 10, 12, 13 years old, we will need to arrange, you know, the blood drawing somewhere close to you. But I don't believe that the company that helped Ms. Behrens and her daughter necessarily can do younger children. But we'll find a way to locally that's safe to get the blood drawn, to help you get the blood drawn, and to get us the blood sample. So, Ms. Behrens, thank you so much for your perspective.
You're very welcome.
So, there's a question in the chat. We've met some other parents with 9p23 deletion, most in Facebook groups. Most of them have been overseas. Can we, if we want to, meet others in the US with chromosome 9p23 deletion? Ms. Dixon, do you want to take a crack at that? Ms. Dixon is one of the leaders of the 9p minus network.
Sorry about that. What was your question?
There's a question in the chat about, we've met some other parents with 9p23 deletions, most in Facebook groups. Most of them have been overseas. Can we, if we want to, meet others in the US with 9p23 deletions?
Absolutely. If you're not a member, there's currently an organization that's been around for quite a while called the Chromosome 9p minus network. We have over 800 families worldwide. We're always accepting, wanting new families to join us. We have a very active Facebook group where we can get to know where families are from, whether it's within the US or outside the US. We also have reunions right now. We've only had the reunions in the US. Maybe eventually we'll be able to do those outside. But at reunions, when we meet up, you get to meet all the families in person, which is a really fun experience. Hopefully that answers your question.
So we have a question from in the Q&A about, is this limited to 9p23 deletions? No, our study is not limited to any specific deletion. We're anxious to get all 9p community members involved.
I'm curious, Sierra Dixon, whether the 9p community phenotypic spectrum has changed noticeably as CMA technology is deployed more frequently, and also, you know, we see smaller and smaller copy number variations.
We don't have too much information yet. We just know there are more and more people being diagnosed, but yeah, we don't really know enough about everyone's whole history to be able to, I guess I wouldn't be able to answer that question very accurately, unfortunately.
Maybe I can add 2 cents to that. just looking at Facebook groups and whatnot, the number of people that say we've just been diagnosed and we have a 85 kilobase pair deletion or 200 kilobase pair deletion, that kind of unheard of, right? And yet you're having people show up to these groups because they have been given that diagnosis, which says there is more precision, and surely that's super helpful for a project like this. So yeah, I mean, I think that the ability to pinpoint is definitely getting better.
That's great.
I try to tell all of our families, especially when they are diagnosed by more recent technology, that a lot of what's in the textbooks is is biased because it's from the most affected individuals who then get tested, and also some of those tests were not as sensitive as some of the newer tests. So it's just really important to remember we're all learning, and we're always learning more. And it's really important for all of us in medical genetics to remember that we need to have an open mind, and that there's a lot more variation than we knew about.
Terrific. We're happy to answer any other questions from the participants in the 9P community. I've tried to sort of keep up with the question and answers and with the questions that are coming in from the chat. If there are specific questions and we haven't gotten to them tonight, or you think about them tomorrow, please don't hesitate to email Tori or to email me. How stable is the funding for this research is a question. And so that's an interesting question. And I'm pleased to say that we have stable funding, and one of our research team's goals is to expand that funding. Certainly Dr. Turner, Dr. Mitra, Dr. Dixon, and I are unambiguously committed to being sure that the general medical research community recognizes how important study of 9P is, not only for the individuals in the 9P community, but also for a variety of different diseases and problems. Tishelle, do you want to say just one word about what you recently identified as how many genes there are on 9P that relative to other chromosomes in terms of neurodevelopmental problems?
Oh yeah, so we recently did an analysis looking at different chromosome arms in the genome, and I think this is what you're referring to, right?
Yes.
Yeah, and what was really intriguing to us was that the connections on each of these different arms, of all of them, 9P has the most interaction, meaning that like the biological, counterparts or, interacting partners with these genes are highly enriched on 9P. And we don't see that on as many of the other chromosomes. We do actually see it a little bit on 8P. And I think there was one other chromosome, but what really stood out to us that 9P really had this higher interaction, which kind of was curious to us that perhaps then if they are so connected, maybe that would explain some bit of why different deletions could result in similar phenotypes. So if you delete part of the network on one part of the chromosome 9P, or you delete a different part of it, that could ultimately result in a similar phenotype. Yeah, so it was really intriguing, that observation.
And just one other, no, thank you very much. I think Dr. Turner's observation helps to emphasize that the studies of 9P are going to be important, not only to the 9P community, but to the sort of medical community in general. We are anxious to receive support, financial support for our studies. If you email Tori, she will tell you how, if you are interested, you can help support this research. As you can imagine, the way this research moves forward is by continuing to make discoveries, and making discoveries, unfortunately, does require resources. So please don't hesitate if you would like to help support this research, or if you know of somebody you think might help support this research, please contact us and we will follow up with you or with the individual and put her or him in contact with the sort of relevant people here at Washington University, where Washington University is a not-for-profit organization. So they're in the United States anyway, there are tax advantages to contributing to supporting research for this project. And we would be anxious to facilitate contributions from anybody in the 9P community who is interested in making a contribution. Thanks for that question in the chat.
But we are super grateful too, just for your participation. You're giving us your blood.
Yes, I hope you could understand that between Dr. Turner and Dr. Mitra and the large team of individuals whom they represent, we have a lot of firepower focused on this, and we need participants to sign the informed consents and to provide their sort of specific medical histories and characteristics, and then to provide a blood sample for them to do the analysis. And it's very, very important to move our studies forward. We're trying to reach 100 total participants who give their blood and sign up consent for participation, and who provide their sort of medical records or medical characteristics, medical background. So as Tori said at the beginning, we're making excellent progress in that area, but we still have 60 or 70 slots waiting for members of the 9P community to sign up, and we would be thrilled for anyone's participation, adult, child, anyone who's affected with a 9P deletion or other 9P problem, please let us know, and we'll get back to you very quickly. I think the chat and the Q&A have now been silent for a while. So Dr. Turner, Dr. Mitra, Dr. Dixon, or Ms. Tate, any final closing comments or suggestions or ideas?
I just wanted to echo your sentiments in thanking Ms. Behrens for coming in and just saying how, as a medical geneticist, how sad I am for some of the comments that people in our field have made in the past, and that I hope that we're learning. And I try to teach all of my trainees to think about children's potential and not about their limits. And I hope that message spreads, and I hope that you know how appreciative we are of you participating and in sharing your experiences and in talking about how important research is, because I come from the lysosomal disease field, where parents are participating in interventions that are very risky for their children to benefit medical research, but also for hoping for their own child's benefit. And so I absolutely appreciate what this means. And while I can't live in your experience or walk in your shoes, I have just tremendous respect and admiration and inspiration for everything that you guys bring. And that is why we do what we do. This is what, and I couldn't be any kind of a success if it weren't for the families. You guys just really are the wind beneath us. So thank you all.
So Ms. Behrens, thank you very much. And also Ms. Sierra Dixon, thank you so much. And Mr. Leon Eidelman, thank you so much for all of your advice, support. Let's see, we have one question here in the question and answer. Great. So that's a parent who looks like he's going to be involved with this child. So Mr. Eidelman, do you want to say a word?
No, just it's so unbelievable to have a research team as talented and devoted and so willing to make themselves available. I think we're lucky that you guys have chosen to work with us, and thank you.
Great. Well, we are so grateful to the 9P community. We're hopeful that we're going to continue to have the kinds of research results that Dr. Turner and Dr. Mitra had described. And we hope that for those of you in the 9P community, if you have questions or concerns or other sort of ideas, don't hesitate to share them. We really appreciate your suggestions and ideas and thoughts. And we're anxious to continue to move the 9P community forward with our research discoveries. So again, thank you very much, everybody, for participating, taking time out of your day. Thanks especially Drs. Mitra, Turner, and Dixon, and Ms. Tate. Thanks to Ms. Behrens for her willingness to participate, and Mr. Eidelman and Ms. Dixon for their ongoing support and contributions. I hope everybody has a great night, and we look forward to hearing from everybody who's interested in participating in this research. Thank you very much.